Okay, Veronica, do you wanna start then?
Yeah, I think it's fine. Okay, so welcome everybody to the second day of our summer school, Adapt 2020. Today our guest speaker is Irja. Thank you that you're here. And she did her master at the University of Oslo, and then she did her PhD at the Norwegian University of Science and Technology in Trondheim, I think. And since 2017, she is associate professor and has her own lab. And today she will talk about lifespan and coevolution with other life history traits in variable environments. And so the screen and the microphone is yours, Irja.
Thank you very much. Let's see. I hope you can see the right screen now. So thank you very much for inviting me. I had an absolutely fantastic day yesterday with all the talks and our discussions and everything. So I hope I'll be able to follow up on that and that we have as great a day today. So as Veronica said, I'll be talking about life history traits and co-evolution of different life history traits in variable environments, especially with respect to lifespan. And before, let's see if it's willing to change the slide for me. Not quite. There we are. Yes, before I start talking about the different studies, I just want to acknowledge that this is definitely not something that I did on my own. My main collaborators on these different projects is Hanna Kokko, William Silva and Thomas Haaland on the theoretical work, and then I've collaborated with Martin Lin on some more empirical work, and we also had other co-authors that contributed to this. So I'm really grateful for all these very nice collaborations. So we all have an understanding of what life history is. Especially this audience, I guess. For most, it will involve both reproduction and lifespan, but also things like growth rates and dispersal, and it can involve patterns in changes in these kinds of traits, like senescence. And obviously, more details about how the individual invests in different components of, for example, reproduction, such as size and number of offspring, and how those are distributed throughout life. Similarly, we can think about different sources of mortality. We can divide them in an intrinsic and extrinsic mortality, and we can think about patterns in mortality throughout life, such as senescence. So I like to think about how many of these traits will respond plastically to the environment. And now this is already getting a little bit too much to keep in mind, and also too much to keep at one slide at one time. So let's simplify a little bit, but still remember that the complexity is really there in the background. So environmental variation obviously affects all these life history traits through plasticity. And what I find really interesting is how the environment can vary in so many different ways. And what I will especially focus on in this talk is how predictable or unpredictable the environmental variation can be, and how that affects the optimal trade-offs between all these different strategies. Also keeping in mind that these trade-offs can go between several of these different traits at the same time, so it doesn't have to be a two-way trade-off. And I also try to show you how longevity is also very important for the evolution of the other components, both in the obvious way that, for example, you can't reproduce after you're dead, but also in some slightly less obvious ways. So plasticity is obviously very important in variable environments. And there has recently been a lot of interest in which types of environments we'll select for plasticity. Here I show you one example of this. And both speed, let's see if I can point for you. So both the speed of change of the environment, and the predictability of the environment is important for when we expect plasticity to evolve. And we can think about different forms of plasticity. We can have irreversible or developmental plasticity, where the individual develops based on the environment it experiences under development, and then it stays the same phenotype for the rest of its life. But we can also have reversible plasticity that I represent with this very colorful fish here, that can change its phenotype several times throughout life. And now back to the axis here. Predictability is important. It's only when the environment is relatively predictable that you can use plasticity, but also how fast the environment is fluctuating. But obviously that is relative to the lifespan of the individual experiencing it. So this is really not just a static component, like a static part of the environment. Lifespan is a trait that can evolve just as other life history traits. So this is what Hanna and I were really interested in, seeing how lifespan can co-evolve with these different plasticity strategies. So to do this, we set up a simulation model where we control the environmental variation. The environment can fluctuate relatively slowly or faster. And individuals in this environment, they are all plastic, but they can be only developmentally plastic. Or they can invest more in plasticity and reverse this plastic phenotype several times throughout their life. And in our model, we see that all individuals, they sample the environment when they first experience, and then they adapt to that. And later they can experience new environments, and then they might be mismatched to that environment if they don't invest in sampling and reversible plasticity. And mismatched individuals, they will experience lower reproduction and lower survival. And then plasticity is costly. So these more reversibly plastic individuals, they will experience a reduced fecundity in the current time step when they are changing their phenotype. So this is an individual-based simulation model with evolution in it. And what is it that evolves? It's this plasticity trait, so how often you sample the environment and change your phenotype. But it's also the reproductive effort, so how much do you invest in reproduction now, and that trades off with survival. So these are the results, and there's quite a lot going on here, so I'm going to walk you through this graph here. And each dot here is one population at the end of the simulation. So this is really the outcome of the evolution going on in the simulation model. The colors here indicate the environmental autocorrelation, how fast the environment is changing over time. So the yellow ones here is a very slowly fluctuating environment. On the x-axis the x-axis here, yes, that's the investment in reversible plasticity. So you have the more plastic individuals over here at this side, and the more developmentally plastic individuals over on this side, and there is a continuum in between. On the y-axis is the mean age of the individuals at the end of the simulation, and this lifespan is really the product of both an extrinsic mortality that is not related to the environment, but also the mismatch with the environment, and how much they decide to invest in reproductive effort. So let's look at some of the patterns. What we see is that in the stable environments, then there is relatively little plasticity. I mean, they're all developmentally plastic, but not that much reversible plasticity. And this leads to a shorter lifespan because they often get mismatched with their environment over long times. When the sampling error is relatively low. So they are good at sampling the environment. There is not a lot of uncertainty. Then we end up with a slower life history. They invest less in reproductive effort, and they have a longer life. And interestingly, when the environment fluctuates fast and sampling error is relatively high, then there are really two alternative outcomes. Frequent plasticity and low reproductive investment leading to slightly longer lives, or developmental plasticity and high reproductive investment leading to shorter lives. So I just wanted to show you what this means for the patterns of what could be something like senescence in these different kinds of populations. And here now I look, still each dot is one population, but on the y-axis, I now have the pattern of phenotypic mismatch with age. So down here, there is no relationship between age and the phenotypic mismatch of the individual, while up here, so at higher places on the y-axis, there is a strong increase in phenotypic mismatch with older ages. So on the x-axis, we again have this plasticity frequency. And what we see is obviously that those individuals that update their phenotype very frequently, they keep being well matched to the environment throughout life. But the individuals that don't really update, they become kind of outdated and mismatched with their environment at older ages. And this is especially clear for these populations here, where the environmental autocorrelation is very low, so the environment is changing quite a lot, and they're also not so good at detecting their environment, so their sampling error is relatively high. So if we look more at what this means between populations, then now on the x-axis, the y-axis here, we have the mean phenotypic mismatch in the population. So down here, almost all individuals are relatively well matched to their environment, but up here, a lot of the individuals are not so well matched to the environment. And now at the x-axis, notice that this is the mean age, so a measure of lifespan. And what we see here is actually that in those populations where individuals tend to live for a very long time, they are still quite well matched to their environment. And this is because obviously the lifespan is a product of this match with their environment. So we have this slightly special case in the middle here. These populations in individuals are pretty good at sampling their environment, and the environment is fluctuating quite slowly. So it doesn't matter so much that they are not updating so frequently, but they still stay pretty well matched to their environment. But in these populations over here, we have this increase in mismatch with age. So obviously, we were quite interested in thinking about, is it possible to see any of these patterns in a real population? And luckily, I have a collaborator that is interested in these same types of questions, Martin Lind. And with his team, he let 30 populations of this nematode here evolve in 5 different environments. And these environments, they differed in their thermal variability regimes. So there are 2 constant environments. One is the control environment at 20 degrees, which is what they are well adapted to. And then we have this constant environment at 25 degrees, which is a mildly stressful environment for these nematodes. We also have this slowly increasing environment, so slowly increasing from 20 to 25 degrees over those 30 generations. And then we have a fast fluctuating environment. So it fluctuates what is every other generation at the start of this experiment. In addition, there was this slowly fluctuating environment where I'm not going to talk so much about that. So what were really the outcome of this experimental evolution? Well, we did actually see quite the response in the lifespan of these different nematode populations, and it has been measured both in 20 degrees and in 25 degrees. And if we compare the populations that experienced increasing or really warm temperatures all the time, they tend to have longer lifespans than these fast cycling populations, both at 20 and 25 degrees. So it does seem like these fast cycles leads to a reduction in lifespan as compared to these populations up here that adapt to the warmer environments. And does it really fit with the model? Well, only partially, I would say. But there are a couple of important differences between the model and the experimental setup, because in the experiment there were only 2 states of the environment, while in the model we had this continuous variation. So what I did was that I re-simulated some of these populations, and now I have only the relatively high sampling error, and I also increased the background mortality a little bit. But the environment was now fluctuating between 2 different environments, and not it's the results of this is really not that different from the main models, but we have a much clearer range of strategies when the environment is cycling quite fast between these 2 environmental states. So we find 2 possible strategies, either more plasticity or shorter lives, faster life history, reproduce a lot and kind of sacrifice your lifespan. So obviously, if anything, this is what we really saw in the nematodes. To sum up this first part of the talk, basically what we saw is that the slow environmental fluctuations tend to lead to very little reversible plasticity, and individuals become poorly matched to their environment over time, which selects for early reproduction and shorter lives as a result of that. While in the fast fluctuating environments, we see much more reversible plasticity, and individuals tend to stay updated in their environment and well matched, which leads to slower life histories where reproduction is spread over a much longer life. Or, as with the nematodes, they can also go for the non-plastic strategy, where they just reproduce a lot, and then we have an even stronger response, even more reproduction and faster lives than we see in these slowly fluctuating environments. So I've been talking now about this actually a continuum between developmental and reversible plasticity as the plasticity trait, but obviously there are other components to plasticity. And the reaction norm is really one very important point, and the reaction norm can vary in different ways. We can have different intercepts or elevations of the reaction norm, but we can obviously also have different slopes of the reaction norm. And before I go on to tell you about a model looking more into this, I'm just gonna point your attention to this little corner here in the figure. And this part actually says that we have bet hedging going on when predictability on the environment is low. And bet hedging is really when individuals or genotypes sacrifice some of their expected fitness to reduce their variance in fitness over time. And I'll get back to this, and but I just wanted to point out that most authors tend to see that plasticity and bet hedging as two alternative strategies to cope with variable environments. So what we did in this model that Thomas Haaland was the lead author on was that we had different reaction norms, and I just want to point out exactly how it works in this graph here. We, as usual, have the environment on the x-axis and the phenotype on the y-axis, and note that the mean environment experienced is in the middle here, and the mean phenotype here in the middle of the y-axis. And then we have this perfect match between the environment and the phenotype along this dotted line here. And a reaction norm with intermediate slope can look something like this, and it will have a mismatch with the more extreme environments. And this mismatch here in the more extreme environment, it translates through this fitness function, and here's the phenotypic mismatch along the x-axis. So this mismatch, it translates into a slightly reduced fitness at these more extreme environments. But if we look at a more intermediate environment, the individual will have a higher fitness because the mismatch is not so strong. And then we have this high plasticity, the steeper slope of the reaction norm, or low plasticity. And we can look at how, well, actually we assume in this model that plasticity is costly, and we assume that the steeper slopes are more costly. This, for example, could be the case if a steeper slope requires kind of a more advanced machinery or cognitive ability to be able to sense the environment and change the phenotype accordingly. So we have this plasticity cost here that is higher for those reaction norms that tend to experience lower phenotypic mismatch, so the steeper reaction norms. And if we then look at the mean net fitness that is coming out of this, both the fitness benefits in the different environments and the costs of these different slopes, then we see that actually it's the intermediate slopes that have the highest expected net fitness here, because the costs of plasticity is so high up here. So if we look now, I think that some of you might be seeing where I'm going with this, but I'm going to work through a numerical example to make sure that we all agree. And I'm just going to now compare two different reaction norms. A more plastic reaction norm that really copes quite well with environmental variation, but it pays a high cost of plasticity. And we have a less plastic reaction norm, so a less steep reaction norm slope, and it copes slightly worse with the environmental variation, as I just said, but it also pays a lower cost of plasticity. And then we imagine an environment that changes over time. Here we have mean and more extreme environments. So the environment keeps changing over time. And we expect that the more plastic individuals, they have a potential for quite high, say, reproduction or performance in the mean environments. They are quite well matched to their environments, but then they pay this quite high cost of having the reaction norm that is independent of the environment that they experience. While the less plastic individuals, they also have this relatively high potential in the mean environment. They are also quite well matched to this mean environment. But they pay a much lower cost. So in the more extreme environments, then we can see that the more plastic individuals, they still have a relatively high potential and they still pay a relatively high cost. And this goes for all individuals. While in the less plastic reaction norm, there is much lower potential for benefits here in the more extreme environments, because they are less well matched to their environments, and they pay a lower cost again. And if this keeps happening several times in these fluctuating, sorry, I was going to mention that the arithmetic mean, which is basically what I've shown you in this figure here, is higher, as I've shown you up here, for the less plastic individuals. But if this keeps happening over time, then we can see that this accumulation of individuals over time, if we assume that each of these eggs grow up to be an adult individual that reproduce with the same reaction norm over time, then we can see that the total number of descendants for this more plastic reaction norm is higher, even though the arithmetic mean was higher for this less plastic individual. And this is obviously because the arithmetic mean is not really the best way of approximating fitness over longer time spans. We need to look at the geometric mean, which is actually higher for this more plastic reaction norms. And that's because they consistently perform reasonably okay in all kinds of environments. So they don't have this really low periods. And this is important when fitness accumulates multiplicatively over time. And this is really conservative bet hedging. So what I've just shown you is that high plasticity increases long-term fitness through conservative bet hedging. So that was a numerical example for two different slopes, but obviously we've investigated this quite systematically for different reaction norm slopes. And here you can see how the arithmetic mean fitness related to the different reaction norm slopes. It goes down here, and note now we have the reaction norm slope on the x-axis, and not the mismatch, but the slope of the reaction norm. However, when we calculate the geometric mean fitness for these different reaction norms, we find a different pattern. And you can see here that we have a higher optimum slope. And this is due to the decrease in the variance in the fitness for these steeper reaction norms. So when is this important? It's actually not always, and this is where lifespan and associated life history comes in. So to illustrate this, I can show you the result of a simulation with these costs of plasticity that I've just shown you. But now the individuals had varying numbers of interactions with their environment, so they kind of accumulated fitness over time with different number of time steps. And now the upper line here is just one time step prior to reproduction, while down here we have up to 10 time steps before reproduction. And this is the resulting reaction norm slope. What's on the x-axis here is the grain of the environment. I'm not going to talk so much about this right now. Maybe we'll talk about it later when we talk more about bet hedging in the discussions later today. But I'm just going to skip this, just look over here and think about the number of time steps prior to reproduction. And we can see that when there are few time steps prior to reproduction, reaction norm slopes become steeper. And that's because bet hedging is much more important in these kinds of life histories, while bet hedging is much less important in these kinds of life histories. And this could come about from, for example, lifespan or parity, so how many times do you reproduce throughout your life. So I'm going to make a little bit of a jump. Let's leave the bet hedging behind for the moment and talk about these biological cliffs. In thermal biology, this relationship between the performance and the body temperature of an individual is called a thermal performance curve. And notice that this function, it's not symmetric, it's typically skewed. And this biological cliff have several interesting properties. You can really see that the body temperature that gives the highest performance is often referred to as the optimal temperature, but a drop in body temperature has only a small effect on performance. But if the body temperature is increased similarly, then the individual has really fallen off this cliff, and there will be a large drop in the performance. And in the same way, if we have stochastic fluctuations in the temperature, they will have large effect on the mean performance of the individual. And this is why we often predict that the preferred body temperature of an ectotherm in variable environments should be lower than the so-called optimal temperature. However, I've talked about this in terms of thermal performance curves, but this shift in phenotype, it's not unique to thermal preference. The same principle holds for any trait where the fitness effects of mismatch with the environment is skewed. And in behavioral ecology, we often refer to this as the insurance principle. So together with Will Silva, I investigated how plasticity evolves when mismatch in one direction is more severe for the fitness than a mismatch in the other direction. So we have this skewed fitness function. And what we did to do this was that we really modified the first model that I showed you today, where we had this a potential for updating at different frequencies to also allow for evolution of different intercepts and different slopes of the reaction norm. And just to show you quickly that the lifespan and how that relates to this reversible developmental plasticity axis is really similar to what we found in the first model. You tend to live longer and are more plastic for some environments, and mean age tend to be much lower when there is much less plasticity. So I want to skip on to look at the intercept and the slope of these reaction norms that we find in these different environments. And I want you to focus on this side here, just because this is what's most relevant for what I've been saying before today. So let's look at what happens to this reaction norm here. When the phenotype is updated quite rarely over here, or when sampling error is higher, then the intercept is lower. So we have the intercept here on the y-axis, and this is the frequency of updating. And this is really the insurance principle that I've just shown you. So phenotypes tend to be slightly off what one could think is the optimal phenotype. And additionally, we see that the slope is reduced so that the reaction norm is flatter when we have a less frequent updating. Now we have this one-to-one environment phenotype relationship here, which we assumed in the first model, and then we see what evolved in this later model when we let both slope and intercept of the reaction norm evolve is less steep and lower reaction norm. But if you remember some few minutes back, what I told you about bet hedging is that we expect bed hedging or geometric mean fitness will really favor steeper reaction norms. So how can we really make this fit together? Well, actually, I didn't really tell you the full story from this plasticity and bed hedging model, because this result depends quite strongly on how we distribute the costs of plasticity. As I said here, the cost of plasticity is related strictly to the slope of the reaction norm, while in the later model, the simulation model that I just presented that gave this less steep slope, the costs of plasticity are really each time you update your phenotype. And we have actually done this model here as well for cost of plasticity that scales when you update. And the results we find here is now geometric mean fitness actually does favor less steep slopes. So it could be that this is partially due to this insurance, but also partially due to bed hedging. But I can't really tell you just yet. We're going to have to investigate a little bit more before I can really tell you for sure what's going on here and why the reaction norm slopes looks like this. So to end with a couple of take-home messages. Firstly, I just have to say that life is really complex, and especially in these fluctuating environments that are just partially predictable. Life history trade-offs are definitely affected by the environment through lifespan as a key trait that really tends to modulate both bed hedging and the success of, for example, plasticity of the different other life history traits. And longer life tends to co-evolve with more plasticity, as I showed you in the first models, where individuals tend to update their phenotypes more frequently when they live longer. But actually, bed hedging is more important maybe if you have possibly shorter lives, maybe only one opportunity to reproduce, and then we can actually see less plasticity as well. So it's not really straightforward. So with that, I would like to again thank my collaborators on all of this work and the funding co-authors, and obviously you for listening. Thank you very much, and I hope we have some questions.
Yep. Thank you for the really fascinating talk here. We do have a few questions in the chat. So the first one's from Daniel Elsner, sorry. So he was asking how was the plasticity cost implemented, in the sense that it was linear?
That actually varies quite a lot between the different models. I don't think it was linear in hardly any of the models. Let's see, I think here we have a more or less linear cost that scales with how much you change your phenotype when you update it. But otherwise, we tend to have this more non-linear costs of plasticity. And in the simulation models, it's even more complicated, because we have costs of plasticity both in several parts of the life history, really.
Okay. And he also asks, additionally, how are the relationships between cost and plasticity set? Is that based on biological data?
No, it's not really based on biological data. So we've done this mostly conceptually. I don't think any of the parameters in most of these models are really very natural, but we've tried to explore quite a wide range. And we have explored some different different costs of plasticity as well. The thing is, if we have very different shapes, then we tend to get either just straight one-to-one reaction norms or very little plasticity at all. So to get this pattern where we see that with the arithmetic mean fitness, an intermediate reaction norm is favored because of this cost of plasticity, then that typically happens with these escalating cost of plasticity. But I think that's relatively biologically realistic as well, because to be able to match the environment perfect at all times, I think that would require quite a lot of an organism. So that's our thinking.
Okay, and question from Ed. Yes, in these simulations, is an individual's ability to detect and respond to environmental cues constant with age? I.e.,
Yes.
could aging lead to a change in an individual's ability to accurately respond to cues?
I guess yes, definitely in the real world, but in our models, this is really this sampling error, and that's constant across all ages. And I guess we could kind of force senescence through implementing a reduced or an increased sampling error over time. But we see the senescence pattern even without that.
Okay. Question from Jonathan Henshaw. He says, thanks, Irja, for the very interesting talk. I'm curious about the bifurcation you predict in the type of plasticity. Individuals specialize in reversible or developmental plasticity rather than doing a bit of each. Do you have intuitive explanation of this phenomenon?
I think it is due to the coevolution with especially reproduction. I think that when these individuals are selected or kind of starts evolving towards less plasticity, then it becomes optimal to invest more in reproduction really early on, and then we get this feedback that kind of selects for less plasticity because of the cost of plasticity, and so we kind of end up evolving towards that corner with high reproduction early on and less plasticity, but and then we get this a similar feedback kind of evolving towards the other corner. But I've only seen that this is kind of the patterns that come out of the simulations, so I can't really say much more than that.
Okay, we have a question from Judith Korb. Says, thank you, Irja, for the nice talk, for the very nice, well-structured and comprehensive talk. Just a question about the first model. The environmental change happens during the complete lifespan of an individual or only during a certain period? And what would you think would happen if it changes across life, i.e. first being unpredictable and then later become predictable?
Oh. I haven't really thought so much about that, but I think in a way it could relate to Ed's question earlier on, but the reverse of what Ed suggested. So the predictability of the environment is really almost a set property. So we do this through how well the individual is able to perceive the environment. So we could easily implement this change over time and say that they are really good at detecting their environment early on, or they're really poor at detecting their environment and adapting early on. My gut feeling is that the results of doing that are going to be pretty straightforward. We're going to see more reversible plasticity or updates to the phenotype if they're not very good at detecting their environment early in life. could be interesting to try.
Okay, question from Lulu. Is it possible to use reproductive value as a fitness measure? Why use the geometric mean?
Well, actually, the geometric mean is really something that you need to look at the genotype level. So if we look just at the individual, it's really hard to see these effects of the variance in the fitness. So we really need this geometric mean over many individuals of the same genotype to be able to estimate it. I'm not sure I completely answered your question, but feel free to follow up if I didn't.
Okay. Question from Barbara. So thanks, Irja, for the highly interesting talk. How much is the outcome of the models dependent on the cost of plasticity?
Quite a lot, I would say. I think one of the main things that I've taken out of all this modeling work is that cost of plasticity is quite important, and especially where the cost of plasticity is coming in, because you see here we get really quite different results when we structure the cost of plasticity differently. But we also get some quite interesting results just from matching and mismatching with the environment without really thinking too much about the costs as well. So it's not the sole predictor of the different patterns of plasticity.
Okay, if there's no more questions in the chat, I'll go with one there, and kind of looking at the slightly large picture of these with increasing environmental variation due to climate change. Do you think organisms experiencing this will invest more and more into reversible plasticity?
That's a really interesting question, and it's something that I'm really hoping to get at. One thing that I'm slightly worried about from this is that if we think about bet hedging and adaptations to longer term variations, I would expect that many organisms have not very evolvable reaction norms, and that would make them less likely to change this plasticity over time. What I also saw in the first models is that it seems like plasticity is a trait that evolves after, and it seems like lifespan is really the thing that evolves first. Now, I didn't have a kind of a gene for lifespan, but lifespan is really the thing that changes first. So maybe that, well, I have to say that the genetic architecture in these simulation models is maybe not the most realistic, but I think what is realistic is that plasticity is quite complex. So it might be hard to evolve new reaction norms, but we've seen that they are evolvable in some species. So it's a very difficult question to answer, but I'm kind of worried that plasticity is not really responding enough to the environmental changes.
Thank you very much. So the in one of the figures you showed kind of a medium plastic option. I was just kind of wondering what an organism that had medium plasticity would actually do. Would they just be slower to react than the highly plastic one, or?
Okay, yeah. Well, in our model, they are not slower to react. They are just not kind of fully changing their phenotype. So say it becomes colder, you could kind of gain more fat or more fur or something like that. And they're not really reaching the phenotype that is quite optimal in that environment. So we have kind of in this model, we have this instantaneous change, which is obviously not completely realistic, but the main thing with the less or the more flatter reaction norms is that they don't reach the optimal phenotype.
Okay. Oh, we have one more question in the chat. Is from Lulu, is variance due to bet hedging or I can never pronounce this word.
Stochasticity.
Yeah. Stochasticity.
Maybe you can explain a little bit more what you mean, Lulu.
Okay, so because as I understand that that hatching is introducing more variance in the offspring, or more variance in the genotypes.
Yes, yeah, there are two types. Yes, two types of bet hedging, and the main type of bet hedging that I've been showing in these models is what we call conservative bet hedging, which tends to lead to individuals not being more different. But then there is this more well-known form of bet hedging, which is diversification bet hedging, which is what you're referring to now when say the offspring of an individuals tend to become very diverse or something like that. But these different types of bet hedging have the thing in common that they reduce variance in fitness for the genotype over time. Yep.
Okay, thank you very much. If there's no more questions, then I'd like to thank you again for the really interesting talk, and I'd like to thank all the people who